Okay, so I'll just begin with an introduction of PAASE. So again, this webinar is being hosted by the Philippine American Academy of Science and Engineering. This organization is made up of scientists and engineers initially based in the Philippines and in Manila, all of Philippine descent. Um, but now we've expanded, as you will see today. Our speaker comes from Australia. Okay, so it's 8 o'clock in the morning. So good morning, everyone. Well, 8 o'clock in the Philippines. So let's begin our webinar. So today's lecture is entitled Decoding the Computing Power of Neurons. Our speaker earned his PhD in applied physics from Osaka University in Japan. He did his postdoc at the Riso National Laboratory in Denmark, where their group pioneered the use of multiple foci arrays to produce dynamic multi-beam optical tweezers for manipulating microscopic objects in cells. In 2004, he established a research group at the University of the Philippines to work on ultra-fast lasers and holography for applications in laser nanosurgery, manipulation of cells, and 3D microfabrication. In 2007, he joined the Australian National University to set up a holographic multiphoton microscope for applications in neuroscience. He established the Neurophotonics Group within the ECLAS Institute of Neuroscience and is jointly affiliated with the Research School of Physics at the ANU. Without further ado, I would like to introduce you to Dr. Vincent Daria. Okay, Vince, you may take it away.
Thank you very much, Kathleen, for the nice introduction. And thank you, everyone, for attending this webinar. I'm very pleased to share with you our work at the Australian National University. So just to give you a brief introduction of where I come from. So the ANU, the Australian National University, is in Canberra, in Australia. So Canberra is in a state or in a territory between Sydney and Melbourne, and it's a very small population of 350,000. So this is a bird's eye view of perhaps the center of the city, where you can see the university around here, and this is actually where the government offices are. Our school, the John Curtin School of Medical Research, this is the facade of our school, and it actually is home to a number of Nobel Prizes. And one of the Nobel Prize winners that's quite important for this work is Sir John Eccles, who did his PhD, also a Nobel Prize in synaptic transmission and neurotransmitters. We also have a recent Nobel Prize in physics, Brian Schmidt, who is now the head of the university, is running the university at the moment. And he did a Nobel Prize he earned his Nobel Prize for his theory and expansion of the universe. So, so my talk today is about how we actually try to solve or actually try to understand how our brain works. So most of you would have used your brain already. And in fact, whenever you see something or whenever you touch or smell or hear something, all of those sensory information goes to our brain, and then our brain would process them. And then whenever we try to act on what we've seen or what we've smelled or what we've heard, then we can act on it, like drinking a bottle of a glass of water, for example. We can control our muscles using our brain as well. So that is a very fascinating function of our brain, how we actually perceive things or how we process those information. So that will be the center of my talk. We're trying to understand how these neurons in our brain process those kinds of information. So, so if we were to build build a computer, for example, this is my former life. I used to be an electronic engineer. We can build a computer using basic logic gates. And for those of you who are engineering background, you probably would understand this, that each of these resistors, transistors can build up logic gates, and logic gates can build up some circuits, and for this particular case, memories, and eventually you can build a computer. And if you, in fact, computers nowadays have become smaller and smaller, your iPhone will probably much powerful than this first generation Mac. In order for us to build circuits, we need to know what are the inputs, how does the input actually are organized to arrive at an output, or the input-output transfer function of logic gates. Like, for example, an AND gate will give you a relationship that would look like this. And also, if you want to build circuits like this one's here, you need some tools to understand how are these logic gates organized and building up a computer. So if we were to build a brain would probably, we can replace some of the components. We can replace the components with some kind of ion channels, or these are proteins that build up what we call neurons. Neurons build up circuits, and a hypothetical circuit of this structure here would probably look like this. And eventually can build up a brain, something like that. And unfortunately, it's we don't build a brain. The brain develops by itself, and it's also what we try to do now is try to reverse engineer the brain. We don't really understand how the brain is actually built, and we're just trying to see how all of those components are functioning in the brain. So we're trying to reverse engineer the brain. And in order to reverse engineer the brain, we need to understand what, again, the input-output characteristics of these neurons, which are the fundamental computing units in the brain. So these are individual neurons. Okay, so just to give you an example of the scale of things, the brain, you can study the brain from a larger perspective, looking at a simple device, a device called MRI, where you can look at hotspots in the brain whenever it's functioning, or doing some behavioral studies. But if you dig closer into a certain region of the brain, you can have circuits where you have individual neurons, and this is probably in the order of millimeters. This one's here, and if you look at a specific cell within the circuit, you can look at neurons, which is probably in the order of microns or tens of microns, and each of these these neurons here will be connected with other neurons using synapses, and this will be in the order of less than a micron, about half of a micron. And then if you dig further down, you can, within the cells, you can have some proteins and some of the molecules that actually make this neurons function. So you can see the large scale of things, and it's really difficult to study the brain as a whole because of this large disparity, or this large range of scales. So what we're trying to understand is just looking at a simple region where we look at single neurons. So we are in a medical research facility, and if we were in a biology department, we could actually look at different brains in insects, for example, or some simpler brain structures. But if in a medical research facility, we need to associate our research to something of medical applications, or for drug development, for example. So we are now we look at the brain of a rat, which is the simplest mammalian brain that we could actually study. And we're looking at a region of a rat brain called the cortex. So the cortex is, there's many other regions in the cortex, in the brain. So the hippocampus around here, the cortex around these regions here. The cortex gets information from different sensory um, from our senses. The visual cortex is somewhere around here, and the auditory cortex and other regions of other senses are also processed in the cortex. The memory is mostly short-term memories processed in the hippocampus. So within the cortex, we can look at several cells. This is a drawing in 1905, a realistic drawing by Ramon Cajal. You can see that the cortex, this is the surface of the brain here. And if you go down further down, this is like a half a millimeter. You can see really large cells and so also small cells. If we were to make a hypothetical circuit for this brain structure, we can have a large neuron, something like this one's here, which is what we call layer 5 neurons. And there's also smaller neurons here that are also integrated. These are layer 2 neurons. What I'm going to talk about more in this webinar is to looking at a specific neuron called this large neuron here, this layer 5 pyramidal neurons. We wanted to understand how these neurons are functioning, so how the inputs are organized for this neuron to fire an output. So and that's the main research question in our group. And we're looking at it, the piecemeal structures. We're looking at certain dendrites in the basal, looking at certain dendrites in the proximal obliques, and as well as distal dendrites here. So again, if you look at, this is the surface of the brain, you go inner down towards the inner part of the brain is this one's here. And the output is actually going through this axon. So a neuron receives input through its dendrites, and it sends an output to its axon over here. We're also interested, given that they receive inputs, we are also interested in how the information is flowing. So if you receive an input somewhere around here, how this information flows down towards the neuron and how that information drives a neuron to fire an output. So those are the things that we're trying to understand. How does information propagate along the dendritic trees? Okay. So, and perhaps later on, as I go along through this talk, you will probably see, understand how these neurons are actually operating as computing units in our brain. So our approaches to reverse engineer the brain, we're actually looking at a lot of experiments. And the experiments we do is what we call in vitro optoelectrophysiology. So we don't have a live experiment. It's called an in vivo. So we have a live mouse in that case. In our case, we call the mice and harvest the brain and look at slices, brain slices, and we call electrophysiology. So we place an electrode into these neurons, and then we try to understand how they function. We also use light, we call it optical stimulation, which I'm going to talk about in detail in this talk. And we also, to stimulate the neurons, and we also try to use light to record the activity of the neurons. And in order to guide our experiments, we also do a lot of modeling, trying to look at how they function from a computer's perspective, and then based on those computer modeling, we can then perform experiments. Since we're trying to understand how the neuron actually also function, it'll be very helpful if we are able to change the morphology of the neurons by cutting the dendrites and see how the neuron actually behaves. What is the importance of that dendrite and its function. Other ways our other technique to reverse engineer the brain is actually to grow neurons on a chip. And I've given this talk in the 2017 APAMS meeting in the Philippines. But this is not the topic of my talk now. I'll be mostly discussing this upper part here. All right. So neurons send information via synapses. So a synapse, so these are, this is the neuron again, and then each of these synapses are actually in the dendrites. A synapse would look like this. This is the sending neuron, and this is the receiving neuron. And for this particular case, this green one is connected to this large neuron here. Once you receive a synapse, the neuron changes in voltage. Okay, so you would know that the neurons are actually separated by a membrane. So you got the inner part here and the outer part. The neurons would have ions that maintain its function. Whenever you have more positive ions inside the neuron, you get a change in the voltage. And this is as a function of time. So in principle, when the neuron is active, the number of ions that goes in and out of the cell changes. And that you can actually measure as a change in the voltage. So if you measure using a voltmeter, the inside of the neuron versus over the outside of the neuron, then you can look at the potential difference between the membrane. So so in order for neuroscientists to understand how the inputs are actually sending information to neurons, you can practically use an electrode to do that. You can deliver neurotransmitters, or you can patch directly into dendrites, or there's many ways to do that. And this is a traditional way to use electrodes. And you can also use an electrode to monitor the output from the soma. So this is the cell body. And again, these are the dendrites, and this is the output, this is the axon. So depending on the number of inputs, you can see the response here. And normally a neuron sits on minus 70 millivolts. And if there's a change in the ion concentrations inside the neuron, it changes in voltage or in potential. If you were to do this experiment, you can have several electrodes. You got a neuron, very small neuron inside your microscope around here, and then you get a lot of electrodes here. And our neuron receives around 10,000 inputs in order to function. So it's very difficult to simulate the function of neurons if you were to use an electrode. And note this is the tip of this one's here is around 1 micron. Any change, or if you tap the table, this electrode will get dislodged, and in fact, it's a very difficult experiment. So what we do in our group is actually to, instead of using electrodes, multiple electrodes, we have only one electrode that's attached to the neuron, and then we use light. Okay, so there's light can actually stimulate neurons, and there are many ways to do that. The basic principle of using light to stimulate neurons has been a revolution in neuroscience at the moment. So it's what we call optogenetics, where they try to encode light-sensitive proteins inside the neuron, and when that neuron, when the protein absorbs light, it opens up and allows ions to go in, and it changes the potential again. So you can see that as a change in the neuron's potential. Another way is actually to use what we call a chemical approach, where you cage a neurotransmitter, and these are proteins that are endogenous or already existing in the neurons, and these are accepting neurotransmitters. And then when you uncage a neurotransmitter, it becomes an active neurotransmitter, and again allows ions to go in, and then you can measure the voltage again. Physicists have been involved in neuroscience research for quite some time already. And in fact, a lot of other techniques have been proposed to use light to stimulate neurons. And you can either directly heat the membrane, and again, change the porosity of the membrane so that ions can go in and out. You can use a nanoparticle, you can heat it up and then change the porosity of the membrane, or you can use a magnetic particle, change the change the magnetic properties of some ions, and then allow um, ions to go in, or you can use photo switching molecules. So these are a lot of techniques, and what we're using in my group is this technique here called uncaging. So we develop a technique called two photon stimulation. The technique is based on holographic projection, which I will discuss later. But in order to do that, we need to have our sample. So this is a brain slice coming from a rat. We call the rat in the morning and harvest the brain, and then slice that into 300 micron thick slices. So this is an example of a slice of a brain slice, exposing the cortex. And there will be a lot of neurons here. And then we just look at one neuron, for example, like this. We then put a dye into this neuron and then do the two photon imaging of the neuron. So in two photon imaging, I won't go into details of two photon imaging. It's been a very established technique, but you can do a three-dimensional reconstruction of the morphology of the neuron. So you can see how the dendrites are going in 3 dimensions. You can look at the field of view this is a really large neuron. This is like half a millimeter, but then our field of view is like 100 microns, and we can see different regions of the neuron here. In fact, we can see specific dendrites that we are interested in. Once we establish the 3D morphology of the neuron, then we can use our uncaging technique. So in two photon, this is two photon imaging. This has 3-dimensional resolution. So we can also use two photon absorption to release the neurotransmitters. And then this neurotransmitter then binds to these proteins into the cell, then allow ions to go in. So that's the basic technique. So at this particular region, we can stimulate the neurons using this two photon uncaging technique. So since neurons receive multiple inputs, it'll be nice to have multiple spots around the neuron. And how do we do that? Okay, and this is where I come from. My expertise in holography and optics brought me to the field of neuroscience. And in fact, when you have most of these inputs, then you can record the activity of the neuron as a function of time. All right, so how do we do the multiple spots across the neuron? So you probably would have seen this movie, a snapshot of Star Wars, where you've got a holographic projection of Princess Leia. Here you can see that this is just a light field, and you can see some bright spots and some dark spots here. In fact, this is a product of a hologram that's placed here and a laser probably inside. And a hologram, you can change it as a function of time. That's why you can see it moving. And that's practically how we do things. A basic principle in optics, or for the physicists around here, you would have light coming in. If you have an aperture, if this is a laser, that means it is monochromatic, just one color. This laser will produce a pattern in this area. You can have maximum constructive interference, and you have destructive interference here. So when you have this constructive interference, you get bright patterns, and if you have destructive interference, you have no light, like dark regions. If you make the pattern more complex, you get a more complicated light pattern here. This is like a two-slit experiment. And you get sharper peaks. And the beauty is that if this one here, this is a fixed pattern, what if we make this one programmable, or something like a programmable hologram? Then you can provide different regions. So you can calculate what the function here to produce a pattern at this region. You get maximum interference here, or constructive interference, and you get destructive interference in these regions. So this is a basic technique. So we can practically, this is from actual experiments looking at focal spots, and it's actually forming the word ANU for um, for ad of the university. The beauty of these holographic projection is that the spots are not contained within a certain plane. You can actually put the spots anywhere in 3 dimensions. So, so you can put the spots in the Z as well in the XY. So, so you got multiple laser spots using a programmable hologram, and you can use a computer to calculate the hologram, and then you can change it as a function of time. You can. So this is one of the first experiments when I joined the ANU, and I tried to do 2 photon this is just fluorescent beads, and I tried to make them fluoresce using multiple spots. This is 25 spots. This is 10 spots in X. Sorry, this is supposed to be XZ, and this is XY here. So this is looking at it from the side. So the laser is coming from this. So this is looking at from this one here is looking from the top. So you can have full 3D positioning of the spots, and the next step then is to use those spots to excite neurons. This is a dream experiment. Of course, our laser has a finite power, so you can practically do so many spots like this. But eventually, we can look at certain regions where we can actively study different parts of the neuron. So holographic projection is before it became popular in neuroscience. I was using it in what we call optical tweezers. But some of my colleagues in the optical tweezers community also brought the technique to neuroscience, and this was one of the first publications using holographic projection by my colleague, Valentina. Their group in Paris published this in Nature Methods in 2008. This is again from my colleague in Italy, Tommaso, and this is our relatively recent paper 3 years ago, where they use holographic projection also to look at the activity of a live mouse. Okay, so at ANU, I built the system when I built up the neurophotonic group. This is the microscope that I slowly built through the years. It has evolved now to more functions. So this is a standard microscope, a DIC microscope, where we hooked a 2-photon microscope, and these are our scanning mirrors. Our holographic projector is this one here. The important thing is that we got a specific device called a DMD, which I'm going to discuss later. So this is the programmable hologram called what we call a spatial light modulator, on SLM. So we've got laser coming in. It illuminates the SLM hologram, and we've got also a laser that goes through here, where we use for 2-photon imaging. These are combined. These 2 paths are combined to image neurons at this region, and this is where our electrode goes in, and we can record activity using an amplifier, and then we can hook it to a computer. One of our first publication of using 2-photon stimulation was in 2012, and it has a number of publications have been published using this microscope through the years, up to last year, in fact. So, so just to give you an idea, this is how the principle of phase-only holographic projection. So you got a hologram. If the hologram looks like this, you've got 2 spots along the X direction. If your hologram looks like this, you've got 2 spots. If your hologram looks like this, you've got 2 spots along the Z direction. If your hologram is like complicated, you got 4 spots, something that would look like this. You can build practically 10 spots or 20 spots here. Some of the spots here are out of focus, and as you increase the number of spots, the power per spot decreases as 1/n. given by this relationship here. So that's basic principle, just conservation of energy, where the amount of power that you will bring into the system is the same. But if you split the laser into multiple sources, then the power gets distributed to the number of spots. So my first PhD student at ANU, Anne, she's now currently at Imperial College. She did the first work that was published in 2012. Um, so we practically, I've talked about a DMD. The DMD is a digital micromirror device. It is actually a set of very small mirrors. If these are the multiple focus or multiple foci coming from your holographic projector, you can switch them on and off, and that's why we can actually do this experiment here. So the SLM is actually quite slow, so you can't really do a high-speed switching. So what we can do is we can switch them very rapidly to emulate maybe synaptic connections, which I'm going to discuss later. One of the things that also makes this DMD useful is that experimentally, when you produce a hologram, you have these unwanted spots as well. So this is what called higher order laser spots, which you don't want, because I mean, there's a finite, if you shine light, there's laser coming, gets reflected from different edges of the device, and these are all unwanted. So you can actually use the mirror device to only shine this into your sample. So this one is removing these parts here. The digital micromirror device can also accommodate different Z locations. So this is at the focus, and this is out of focus by 10 microns. This is in focus at plus 10 microns. The nominal focus is at 0. So you can, again, produce, this is how it looks like when it's in focus at plus 10, and this is out of focus at minus 10. And if you change the reflection sample, you can see it around these regions here. Oh, sorry, focus at minus 10 and out of focus at plus 10. And the mirror, the mirror setting is actually looking like this one here. So you get larger mirror setting to accommodate for a broader light that's coming from out of focus beams. So using that technique, we can produce spots anywhere within this region here of a neuron. These are 5 spots, and we can randomly turn them on and off using the digital micromirror device. And what we've seen is that once at this for this particular experiment, you need at least 4 spots that are simultaneously turned on to make this neuron fire an output. This is what we call an action potential. All of these outputs are what we call action potentials that are a product of a neuron receiving inputs from these regions here. All these outputs are measured through this electrode, and the inputs are set by the light or the multiple foci. This experiment tells us about how the distal outputs are correlated with proximal inputs and how they actually result to an output. Okay, so again, we're trying to understand how the inputs are actually organized for this neuron to fire an output. This is one of the first publications we got from the microscope, which got us more collaborators. In fact, we've collaborated with a neuroscientist group in Germany at the University of Regensburg, where we built the system, the holographic system, into their microscope. And this is how it looks like. And the person who did the experiment is Max at the University of Regensburg. But then the guys who helped them was Michael, my current my PhD student, and Marianne, who used to be my PhD student, who's now at Imperial. So we helped them build the microscope, and we performed this experiment, which was published last year. we try to place inputs into a specific set of dendrites. And in fact, these inputs are actually in 3D. So if you see these dendrites, it's actually rotating. It's not in a single plane. So some of the inputs are at different planes here. These are two inputs. These are five inputs at one plane, and these are three inputs at another plane. Each of these inputs will actually give an output, will be recorded by this electrode. These are the recordings for input one. This is a recording for input two. This is again the spine where the synapse would actually occur. And for this particular case, we don't want to wait for the synapse to receive inputs from another neuron. We're just using light to stimulate the input, and so on and so forth. So you've got 1 to 10 inputs, and these are, again, this is probably sitting at minus 70 millivolts, and this is about 1 millivolt change. Once you turn them one at a time or simultaneously, or one or 1 2 simultaneously, 1 3 simultaneously, you get this different responses. And if you turn them on all together, you finally get an output. So for this particular experiment, you need at least 10 inputs to drive the neuron to fire an output. And this is at the dendrite, at this particular region called the basal dendrite, and these are the input organization. We can look at different dendrites as well and see how the neurons or dendrites are responding to that many inputs. And we've looked at several. If the inputs are linear, so if they just add up all together, it will follow this line. But apparently, as you increase the number of inputs, the response is actually nonlinear. So those are the interesting things that would probably matter in terms of computation, whether the inputs are linearly summing up to set up an output. Most of the dendrites are actually what we call nonlinear properties, where they need maybe 5 or 10 or some inputs, or maybe 20 inputs, depending on the active properties that are inherent in that dendrite. So, okay, so what I've just shown you was that our capability to use light to stimulate the inputs, and we can record the outputs here. So, and again, the recording is in voltage. It will be nice if a particular input here, we can also see what's going on at this specific set of dendrite. And in order to do that, we need to put an electrode here again, right? So if we were to probe the activity of this dendrite when this one is setting an input, we need to place an electrode. So the way to do that is actually not to use, again, electrodes. We use light. And we can use light, again, if we place the neuron with a calcium dye or a voltage dye, when calcium is one of the active ions that goes into the cell whenever it's inactive. So whenever the neuron is firing, there are more calcium that goes in. So that actually, calcium is a positive ion, so that actually increases the voltage of the neuron. So if there's a dye that actually monitors the amount of calcium that is inside the cell, you can then look at the fluorescence of that dye when it binds with calcium. And then if the fluorescence changes as a function of time, then you can correlate that with the voltage. It's not an exact correlation, as you will see later, but you would get a sense of whether the neuron is firing or whether the neuron is active or not. And if you place, again, the same number of spots anywhere within the neuron, you can look at the changes in the fluorescence. Okay, this is the change in the fluorescence as a function of time. So we can then use this technique. So instead of using it to stimulate, we can use optical recording of neuronal activity. using calcium imaging. This is just the overview of how much calcium. Again, this is inside the cell and this is outside the cell. And these are some of the proteins that allow ions to go inside the cell. Once a neuron is active, ions go in. Don't worry about the name of the proteins. If you're a neuroscience biologist, you would understand that. But in principle, ions goes in, and one of the major ions that goes inside the cell is calcium. If this is an action potential here, the calcium goes, um, uh, this is the amount of calcium, this is calcium concentration here, and then it goes as a function of time, follows like this. So calcium goes in, and then calcium goes out as well. This is a simple experiment where we did in the lab. We just place some kind of chemical to make the neurons active. And you can see that the fluorescence change as a function of time, right? See, you can see here, even the dendrites are communicating, it's sending information to the different neurons. This is the cell body. And if you plot the fluorescence as a function of time, you can look at how they actually correlated to each other, how they are sending or how they are communicating as a function of time. And if you have that kind of tool to analyze the brain, you can look at circuit activity. And a lot of people, a lot of neuroscience experiments are based, recent experiments are based on this technique. What our lab is using this technique is to understand, again, going back to my main question of trying to understand the input-output characteristics of neurons, we're looking at a certain phenomena in different dendrites where they act, what we call a dendritic spike occur. So an action potential is a spike that is formed at the axon or the soma. So you get action potentials, very, very sharp spikes. But in the regions, in the dendrites, you can also what we call a spike, what's called dendritic spike, which is not quite similar to an action potential. So if you place inputs into the neuron, like this one's here, this neuron will change in polarity. So this is, again, the color is a change in the voltage. It will send information from these regions here towards the soma, something like that. And then eventually it will fire action potentials. If you plot that as a function of time, it will look like this one here. So this set of inputs drives the neuron to fire these two outputs, these two spikes. But before it did fire these spikes, it produced a dendritic spike at this region, which is a lot broader. In fact, it's the voltage is a lot higher. So this has been research has been going on for a long time. In fact, this dendritic spike at this region has been proposed since 1999, and what we're trying to understand initially was how this dendritic spike is actually setting the set of inputs to produce an output from this neuron. Another way to produce dendritic spike is that if this one is firing action potentials, the action potentials actually back propagates to the dendrites. And if you have a series of action potentials, like 4 action potentials, you can also drive a dendritic spike. So, depending, if you look at information that goes into a highway, so if you look at this main branch here called the apical trunk as a highway, you've got some roads going in. So if you count the number of cars going through a highway, some of the cars would go into the side roads. You end up lesser cars here. And that's what we originally wanted to understand, is how this set of dendrites are actually impacting the flow of information. In fact, if you fire action potentials, something like this, 4 action potentials, it drove this part to have dendritic spike, similar to what you've shown earlier. But the set of dendrites here are actually also getting some information, or getting some voltage due to the back propagating action potentials. So the question that we're trying to understand is how does this set of dendrites are actually impacting the flow of information, or can these dendrites evoke dendritic spikes itself, not just in this region? So that's what my recent PhD student did for his work. Michael is actually in the audience. He did numerical modeling to look at dendritic spikes first, um, and look at different regions. So in a computer, you can place an electrode everywhere because there's no physical limitation. You can practically model everything, and you can place an electrode anywhere you like. And you can look at the voltage, how it changed, as long as you have a realistic model. For this particular case, he was able to model the dendritic spike at this region here, where you get this large spike only at 100 hertz spacing between these action potentials. If you have 90 hertz or 1 over 90 hertz spacing or 90 hertz frequency of this spike train, you don't get anything. So you sort of get some kind of switch where it's a frequency dependent switch. Below 90 hertz, you don't see a dendritic spike. And if you have above 90 hertz, you get a dendritic spike, something like this. So you produce this only when you meet certain frequency to fire a dendritic spike. What Michael has shown is that he was able to see a similar kind of dendritic spike occurring at this region here, at what we call oblique dendrites. And this is the same, this is the oblique dendritic spike that he was able to observe numerically first. And it has the same characteristics. Below 35 hertz or 30 hertz, you don't see anything. At 35 hertz, you do see something. So it's a much lower frequency compared to that. And it goes similar pattern that would look like this one here. So this was in the model. So to verify that model, again, we use our technique, the multi-site 2-photon imaging, to look at different regions of the dendrite. And we can look at dendrites here, and we were really interested in the set of dendrites that I just mentioned earlier. So this is how it looks like from if you look at the different planes of the neuron, and if you place your spots, these are the spots. Let me see, just will come in later. Yep, these are the spots here, and if you look at the flicker of those spots, you can see how it's affected by back-propagating action potentials. So we introduced the action potentials using the electrode. Now we're not using the electrode to record activity, but to drive the neuron to fire action potentials. So we're putting current in, and it's firing action potentials, and then we're recording the activity from the dendrites. So that experiment actually gave us that the model is actually correct. We can see a dendritic spike at these yellow regions here. The red ones we don't see, so that doesn't happen everywhere. Most of the red one here is at the trunk. This is a trunk here. This is a dendrite, but then it doesn't happen in all dendrites. It happens on specific dendrites. But in a dendrite, it's occurring, it actually produces a very nice, what we call dendritic spike. This is a measure of the calcium whenever you have two spikes. And if you plot that, the step function again as a function of frequency, you see that it has again a frequency response, a step in the frequency response. Other dendrites doesn't have, but for this particular set of dendrites, these yellow ones here, it will have almost the same profile as this. Okay, so there's a big jump in calcium for this specific set of dendrites that Michael is looking. Again, we can put a curve, we can fit the curve by a sigmoid function, or that would represent a step function. Aside from the fact that you can see the response of the dendrites, you can also record activity from the soma. So it's what we call after depolarizing potential. So this is the model. So this small bump here, you can see it measured by this electrode. This bump here is also measured by this electrode. From experiment, we can look at that. Again, this is the measured response from this electrode here. Below 40 hertz, you get nothing, and you get above 60 hertz, you get this bump here, which is at this region here. So again, we see that also in experiment. If we plot the after depolarizing potential, or the ADP, you see this step function, which is nice step function for some neurons. Some neurons actually have this kind of two-step function. This is actually brought by the dendritic spike at this region here. But this one here is specific for this set of dendrites. Again, so we know that the dendritic spike that we found numerically also exists in experiment. And we've done that using the technique that I just described. But what if we cut the dendrite? Can we actually observe changes in the neuron? So during Marianne's time as a PhD student, or a few mistakes ago, we actually developed what we call a laser dendrotropy technique, where we try to cut dendrites, and we've done that systematically, trying to understand how the importance of different dendrites and its functions. And this was published about 4 years ago, and actually meticulously cut all the a certain set of dendrites, and see how it affected the function of the neuron. So once you cut the dendrite, the neuron is still alive. It seals back after the region where it was cut. It seals back within 5 minutes, and then you can see the neuron functioning again. But it functions differently after cutting. So, and in fact, this paper that we published in 2016 describes all the details and how to keep the cells alive after cutting, or I mean, of course, if you shine too much light, then the cells gets dead. But you need to keep the cells alive after cutting. And Michael has applied the technique to study the dendrites. And in fact, the step function before cutting the dendrites is cut a certain dendrite here, and after cutting, this step function was gone. The cells is still alive because it's still firing action potentials here, but then this bimodal changes in the ADP, you don't see that after cutting. So again, this is again a proof that this dendritic spike occurred at this specific set of dendrite that was cut. All right, so what are the implications of what I've just mentioned, or I just talked about? Dendrites, or the neurons, I just mentioned, are important for processing information. In fact, they're also very important for learning and memory. In a real brain, how do we learn? So if this were a live audience, I would have asked, raised the question, how do you learn? How do you practically learn a skill, for example? And let me just show you an example. So this is trying to play a certain piece, a difficult piece. You see some mistakes, and then you repeat. So practically, you learn something by repetition. In fact, it's common everywhere that if you want to memorize, you probably read that stuff all over again, and then eventually your brain could memorize, or could learn some kind of information that you need to store. Okay, this does the real brain does that. In fact, artificial brains also do that. So the neural networks have connections between the neurons, and the connections between neurons are called weights. And this is actually, the weights are associated to the strength of the synapse. So the synapse is actually connected from one neuron to the other neuron. And this is how we learn. This connections between these neurons actually increases in strength if you have learning. And this has been proven, this has been a dogma for quite some time, is what we call you change the synaptic strength called spike timing dependent plasticity. So whenever this neuron drives, when sends an output and it drives this neuron to fire an output, you've got an increase in synaptic strength or synaptic weights. The strength of the synapse is dependent whether this neuron here is resulting for this neuron here is resulting for this neuron to fire. If this one happens after that is well, this is what happens. So the neurons, they're not correlated. So that the firing of this presynaptic neuron is not correlated with the output of this neuron. But if they are correlated, if this one is driving this one to fire, then they are increasing in weight. And if you do that in repetitions, so these are actually number of repetitions. This is experiment in 1998. You can increase the synapse. And what do you mean by increasing the synapse? It's actually physically changing the size of the spines that are connected. So this is the presynaptic connection, the bouton. This is the receiving region called the spine. And when you have an increase in synapse, it's either you have an increase in the number of channels, or practically this one increases in size. So when this one is increased in size, you got a higher change in the potential. So it makes the neuron more receptive to inputs or to change in the ionic concentrations inside the cell. So when you have, and this doesn't happen in single neurons, it happens on all of the synapses, depending on you learn. So whenever you learn something, or when you see, you memorize a face or you recognize the face, it's practically a memory, a synapse that is inside, that is represented as a strength in the connections between your neurons. And this is a standard example of learning memory. So how does this relate to what we just talked about? Okay, so if this one if this neuron here is sending information to this one fires an action potential, and eventually this neuron will see this just as a blip. Remember in my first part of the experiment where each of the synapse here is just giving us maybe 1 millivolt change, and this is just 1 millivolt change. This is the size, the difference between the size of an action potential and the 1 millivolt change over here. You need quite a lot of inputs to make the neuron fire an output. If you have a dendritic spike, or this neuron, if this dendrite here is already changed in polarization, that would look like this one that I've shown earlier. Whenever you have an input within, or whenever this neuron fires an input within this range, this will eventually, because this is already depolarized, it will, the chances of this neuron firing an action potential is very high. So in principle, the dendritic spike drives this neuron to fire an action potential, and eventually, if it's from the STDP technique or theory, the synapse gets stronger as you repeat it. So dendritic spikes is really helpful to drive inputs to an output. Okay, and this, in fact, dendritic spikes in these regions here in the apical tuft has been well studied. These regions here, but what about these oblique dendrites? Okay, in fact, I mentioned earlier that we tried to look at the study by studying the obliques, how they affected dendritic spike that is propagating. But we found out that actually this obliques also has dendritic spikes. In fact, this set of dendrites have been overlooked by a lot of neuroscientists. If you look at this picture here, this is the trunk. Oh, forget about my bike. What I'm really interested here is that this apical, this trunk of a tree would have oblique branches, which are actually outgrown. And some of the theories that these oblique dendrites are actually old dendrites that has been there when neuron is young, and when they outgrown it, most of the function is actually in the apical tuft, where there are leaves in here, so there more sunlight, but these regions here doesn't have anything. They're just old dendrites that doesn't function anything. So nobody has actually um, brought attention to this set of dendrites for quite some time. So what we've really shown in the work by Michael is that this set of dendrites are not um, have function, and they can actually provide what we call dendritic spikes as well. And in fact, if you, in fact, most of the a lot of some inputs actually go into these regions, and that's important for also sensory information. Um, and in fact, yeah. So what we've shown is that dendrites, this region here, provide dendritic spikes. They can actually help this neuron um, make firing more probable. So in summary, we've shown you a technique called holographic projection, and we've used it to produce synaptic inputs to specific set of dendrites like this one's here, and we can make them fire outputs. We can use the same technique to do calcium imaging, and then we've got also um, um, use it to look at specific set of dendrites in the apical. Okay, just to give you proper context, the dendritic spikes that we mentioned happening in these regions here, the apical top, has been studied since 1999. In fact, they have Matthew Larkham's group had a recent paper in Nature last year that these dendritic spikes also occur in human neurons. So most of the neurons before, most of the studies before were rat neurons, but here I think it was sometime middle of last year they published that it occurs also in humans. My collaborator also published work that these dendritic spikes occur in the basal dendrites. But the new thing about this study with Michael is that these dendrites are also very helpful in terms of function. So with that, I would like to thank my collaborators. The work on dendritic spike was done by Michael. He just finished his PhD. In fact, he's got his PhD approved in March, and he went home as well in March. He's now in the Philippines looking for work, I know. You're welcome to hire him. He's a very intelligent and very skillful guy. He plays a lot of piano as well. Um, Hans has been my mentor for quite some time. He's been, he hired me to ANU. Brian did some of the modeling. Vinny did some of the brain on a chip studies, which I didn't talk about this work. Xiaoyuan is my postdoc from China. He's doing a lot of work with Vinny as well in terms of growing neurons on semiconductor nanostructures. These are my collaborators at ANU, physicists and neuroscientists. And Anne, my first PhD student, is now currently in Imperial College doing her postdoc, and some of the guys who did work on, in the lab, and our collaborators in Germany as well. We got funding from the Australian government, but from the Research Council and the Medical Research Council. Thank you very much.
Thank you very much, Vince. Okay, that was a very interesting talk, and I really liked how you transitioned initially with a background in engineering, moving to physics, and now we're talking about neurons. So, just to remind our participants, for those who are not aware of the chat function, so again, there's a chat function at the bottom of your screen. So, if you have any questions or comments for our speaker, kindly type it in the chat. Okay, we have a few questions from those watching in YouTube. So the first one is from Angeline Lau. So in slide 12 and 13, you show that light is used to stimulate neurons. If light is turned on and off, does it or can it mimic a circadian rhythm of neurons?
Yeah, yes, that's a very interesting question. In fact, yes. I mean, in fact, the frequency-dependent responses of the neurons are almost related to the temporal responses of the neuron. The circadian theory is mostly a systems approach where you have multiple neurons. We're looking at a single neuron we could probably extend that to multiple neurons. And in fact, yes, people have studied switching of the stimulation so that you can probably look at synchronous activity or looking at a frequency dependent activity depending on the frequency of your stimulation. Yes, it can be done.
Okay, thanks. The next question comes from Raymond Tan. So he said, he's curious. Is it possible to draw any insights on how to improve training algorithms for artificial neural networks?
That's again very good question. In fact, in most of my grants, I like to appease a lot of people. My grant applications, sorry, some of them didn't get funded, but and I have to say that the techniques that we develop in trying to understand how the neuron works feeds into new algorithms of trying to new algorithms in artificial intelligence and trying the learning process and how neurons in artificial intelligence can actually behave like real neurons. In fact, in one of my talk about trying to grow neurons and semiconductor chips, we're trying to build what we a sort of neuromorphic device where it's a bridge between a real neuron and an artificial neuron um, to mimic exactly what happens in the brain, but then it's actually you're building it. You're not using an existing neuron.
Okay, thank you. I think Joel has a question for you.
You, Vince, congratulations and thanks for this wonderful presentation. I got a better appreciation of the significant advances that you and your team have achieved through the years. So I'd like to refer to one of your earlier studies where you were firing 10 dendritic inputs to get a neuronal response. And you found that all 10 inputs needed to be active for you to get the neuronal activity. When only two, for instance, were firing, you weren't getting any response from the neuron. But my question is, during those combinations, were you conserving the total amount of energy or power put in so that when only two were firing, it's the same total amount of energy as when 10 were firing?
No, no, we've added energy. So that means we've actually removed
Right.
the light spots that were were out of the picture. So it's
Correct.
reduced energy. So you
So
increase the
my,
energy as
my,
you
my question there was, do you think that there's a threshold, a minimum threshold of input energy that's needed regardless of the locations or the number of
yes.
dendritic inputs are firing for the neuronal activity or response to be achieved?
It's a very good question. In fact, there is a threshold, and it's about a certain millivolts from the resting potential. It's called it's about 40 millivolts, and you need to surpass that.
All right.
That threshold to make the neurons fire, and
Right.
in fact that makes them the neurons nonlinear because of this threshold. In fact,
Right.
it's very interesting before we reach a threshold how we actually um, how the inputs are actually acting up to build up that threshold to reach a threshold, whether they've been linearly acting up before the threshold and eventually get a nonlinear change. And it's primarily because of these proteins that are in these neurons, how they actually, I mean, there are several proteins that are responsible for the ions to go in and out of the cell, and how they you practically tease out which proteins are active at one point in your stimulation. So
Right.
those are the specific questions that we're looking at in terms of
Right.
how these are actually organized and which dendrites actually contain these proteins. Do the dendrites have different protein distribution, or are they uniformly distributed? It actually gives us an idea of whether some of the dendrites function differently from the other dendrites, whether some dendrites are additive or some dendrites are
Right.
multiplicative, and
Right.
whether they actually form different functions towards its for the neuron to fire or not. But that exactly is our question.
And one last question, sorry. What was the wavelength of the laser that you used? And I was wondering if you tried another wavelength for the laser.
Okay, so the laser wavelength that we've used is specific to the absorption of the chemical compound that we used. We're using uncaging, and it has a UV absorption spectrum. To do a 2-photon stimulation for that for that stimulation, we need to do twice of that. So we're using around 720 nanometers. It's a ultra fast laser. It's a femtosecond laser, so it's near infrared to produce an absorption in the UV range around 360 nanometers.
All right. Thank you. Very fascinating. Thank you.
Okay, we have another question from Angie Lau. In slide 37, you showed an illustration of deep neural network. Have you applied deep learning approach?
Not yet. I think Michael, my PhD student, would has some kind of idea before to analyze our experiments or data, because we've got a lot of data and tried to tease out some of the patterns that we've been looking at, but we've never used it as a tool. In, there can be some. I mean, a lot of groups in the world have been using neural networks or artificial intelligence to tease out some patterns, especially if you're looking at really large data. What right now we're just looking at a single neuron, but if you're looking at multiple neurons, if you're looking at in vivo experiments where you're looking at circuits, then you need to really have a very powerful computer and a sorting device that would find patterns in the circuit activity. But eventually, if we were to expand our research to circuits, then that would certainly happen.
Okay, thank you for that. We have a question from Lulu Harold. Can insights in slide 37 and 38 help in the memory improvement for the aging population?
Um, what we've shown in what I've shown in slides 37, learning in memory, is a basic principle of learning in memory. It happens in all ages. The degeneration that happens in aging is when you have loss of memory. It's and there are many research on Alzheimer's disease, for example. In fact, Anne, my current my former PhD student who's now in Imperial, is actually looking at Alzheimer's disease and how it changes, how certain brain regions actually lost some kind of memory. What I've shown in slide 37 is basic principle. It's fundamental principle. Whether it can help research on improving, trying to combat Alzheimer's, I'm not sure. But as a fundamental theory of learning, it could probably guide them on some aspects, but probably indirectly.
Okay, thank you for that. We have a question from Ramil Orshosa. So he's working with ethnophysics, a combination of culture and physics, particularly in hand woven of Tiboli weavers. So is it possible that the weaving designs of the Tiboli weavers are the same neurons activity of their brains? For example, in the arts, linear or nonlinear output could be equal to the neurons activity of a certain person.
I can try to answer your question. I'm not into that field, but it's very difficult to extend what we can do, what we've analyzed from a single neuron perspective, into how the whole brain works, right? So, I mean, I've shown you in at some point a slide that actually ranges from several microns in range, which is around micron to around half of a meter or maybe a fourth of a meter for the size of a brain. So the large discrepancy in in brain sizes makes it difficult um, to extend what we've seen from a single neuron to eventually find it functioning in the whole neuron. So systematic systems neuroscience, when you look at brain patterns, especially regions in the brain where the analytical parts of the brain is functioning and the artistic part of the brain is functioning, that's probably best answered into that area where more behavioral responses um, are associated. But from the fundamental um, part where I talk about single neurons, it's difficult to associate that. But the patterns that I can only speculate on the fact that when you're good in arts, you could probably embed that into your brain, and whether the patterns that are in your brain, they could be synapses that are stored in the artistic part of the brain. There is a lot of work looking at what we call memory engram. So an engram is actually how the synapses are actually formed in specific circuits in the brain. But they can do that. They can do that in rats, for example. I'm not sure. They haven't done that in humans. They're looking at how a certain memory is stored in the brain in terms of synaptic connections and so um, and since they can only do that in rats, they can only do that in simple um, behaviors of rats, like maybe finding, navigating through a maze, or navigating, looking for food, and something like that, and not from the artistic point of view.
Okay, thanks. We have a question from Edsel Peña. So it was a very nice talk. How will it help understand how humans make decisions? Say, for example, I have an apple and a banana. How do I make a decision which one I will consume? And how does my brain, or how do the neurons process this particular problem?
Well, it's a very good question. In fact, in one of my introductions in several mistakes ago, um, uh, I was actually relating that the neurons receive multiple inputs. It means it receives multiple information simultaneously. Whether the neuron would fire, it does not. It does not fire only from a single train of thought. You get multiple train of thoughts. So if you were to select a lot of things between two objects or two decisions, you get information from several sources in your brain. You get information from your memory. You remember that it's actually good that the apple tastes better than the banana, but then the banana actually looks more fresh, something like that. So you get new information from the freshness that comes from your senses, but your memory tells you that the other one tastes better. So in a way, once all this information gets processed by a neuron, um, you then the neuron tends to decide, and whether that translates to the brain functioning, it's still difficult to make a conclusion out of that. But in a way, what we're trying to do is that we're not making the neuron just receive inputs from a single source. We're trying to understand a holistic point of view where a neuron receives multiple inputs.
Okay, Edsa has a follow-up question. Do you think that we are using some form of evaluation function?
Another very great question, and I don't think I can answer that. And that's one of the things that's just probably really interesting in this work, because there's so many questions that you can actually ask, whether and based on our experiences. What we can show from the rat's point of view, from the rat brains, is that they can navigate, they can smell, they can see things, they have a visual cortex as well, and whether all of those uh, information goes down to a single neuron or multiple neurons, whether each neuron would actually eventually find the proper way to process that, it's still a very big question in neuroscience.
Okay, thank you for that. We have a question from Al Serafika. How did dopamine affect the neurons in the movie Awakenings, causing the catatonic patients to dance again?
All right, so dopamine is again a form of neurotransmitter. It's like what I've shown you earlier is a neurotransmitter called glutamate. Okay, so dopamine is one of those families that actually activates a protein that would make the function that would make the neurons receive ions. Okay. Um, there are many other neurotransmitters like serotonin, and I think dopamine is mainly. I try to answer your question. I'm not a neuroscientist, but then from my basic understanding is that um, it uh, responds to pain. I think from from my memory. I'm trying to use my brain to understand it. I don't know what happened in that movie. I haven't seen that movie, but then it could affect some of your brain function whenever when you block a certain protein to respond to dopamine, for example. So if you practically change your neurons not to receive dopamine, then it could affect your behavior. Similarly, if you block pain relating proteins in your neurons, you don't feel pain, and that's what practically you would still have a scar, but then your brain will not feel any pain because of the pain relievers. And that's a similar function of trying to manipulate neurotransmitters in terms of behavior. Yeah, I'm still on. Hello.
Hello, Vince. Sorry, I think I lost you there.
Mhm.
Okay.
Did I was I
Um,
caught?
y- yeah, I think yeah, can you yeah, I think you were talking about yeah, how the dopamine can
Can change the behavior.
yeah.
So it's again as I mentioned, it's like a pain reliever or that blocks a certain proteins to receiving some kind of neurotransmitter, and it changes your behavioral responses.
Okay, thank you for that. Um, all right, I think we've gone through all the questions. So maybe I'd like to ask you, what's the next step? Like, what are you up to now? Like, what goes on after this study, or what are you working on right now?
So prior to COVID, our the neuroscience research has been really exciting. I don't know after COVID if we still get funding to do our research. I'm practically it's been it's over 10 years since I've built the lab, and my PhD. I don't have a new PhD student at the moment, so I'm probably closing my lab and moving to physics, but still collaborating with neuroscientists. So that's some of the plans that I'll be doing in the near future.
Yeah, it was very interesting. Okay, I think, do you have any other words for our viewers?
So there's one question.
Oh,
I think
hold on,
but
let me
it
check.
was sent privately.
Oh yeah, would you like to address it?
So okay, thank you. Neurophotonics is a very interesting field. Can someone from other fields of science go to this field or PhD study, perhaps? Yes. In fact, a lot of engineers. Neuroscience is a multidisciplinary um, very multidisciplinary topic. We've got chemists, we've got biologists, we've got physicists, we've got engineers. is actually giving a lot of technology, building microscopes, building amplifiers, building electronics to read neurons, read the brain is cannot be done by one field of science. It's very multidisciplinary. So yeah, you can start to be an engineer, and in fact, a lot of topics in bioengineering is actually very helpful in this field.
And I think you are a testament to that, being able to cross different disciplines and
So
establish your
I
own
was
expertise.
telling Kay before my talk that before I came to neuroscience, before 2007, before I came to ANU, I knew nothing of neurons. The most the most experiments on cells I've done was on yeast cells, and that's why I can communicate with Kay about making bread, because I've been looking at yeast cells for some time before.
All right.
Okay, Giselle sent me a message that she has a question. So while we're waiting for that particular question, maybe I'll announce first our lecture for next week. So please, okay, so I hope you can see my screen.
Yep.
Um, Okay, so we have our lecture today, and next week it's going to be our 10th lecture. We're going back again to COVID 19. So it's entitled Policy Development for COVID 19 Pandemic Response Using System Dynamics. So it's going to be on June 5, again at 8 in the morning for those in the Philippines. So our lecturer is Dr. Charlie C., who is an associate professor and research fellow in the Department of Industrial Engineering of De La Salle University. So she obtained her PhD in Industrial Systems Engineering from the National University of Singapore. Together with Charlie, she will be joined by academician Alvin Kulaba, who is a known expert in engineering systems modeling. So if you're interested to join that webinar, please register at bit.ly/CharlieC. So that's CharlleSy. Okay. Um, so again, oh, okay. This is the, hmm, okay, I'm not sure. Okay, I'm not sure about the question. Okay, anyway, so for those who would want to follow PAASE and its activities, the Philippine American Academy of Science and Engineering has its Facebook page. Um, and it also has its website at paase.org and all our webinars. So, in case you didn't catch any of our webinars but would want to listen, they are uploaded in YouTube at bit.ly/paase webinars. Okay, now I have the question from Giselle. So she said that they use calcium fluorophore imaging to see how conopeptides affect pain and neurodegeneration receptors. Would you have any comments or insights with that?
Yes, calcium imaging has been widely used in the field of neuroscience. In fact, um, the cone um, the shell cone research by Giselle that actually provides a good pain relievers goes through the brain as well. So I mean, I mentioned earlier that whenever you feel pain, you just drink a pain reliever, and that the cone research done by Giselle and Baldomero Oliveira's group and has been really important in neuroscience, because they practically all the information goes to the brain, and how the neurons actually deprive feeling deprive the animals from feeling pain. Yeah, different areas of the brain regions have been studied, and calcium imaging, you can what I've shown you is that a dye, you can practically use a dye to do calcium imaging, but a lot of work now has been using genetically encoded calcium indicators. So you can look at a live mouse, and these are these fluorophores. Not just they're not molecules anymore; they're incorporated into the proteins of the animals, and genetically encoded into the animals. And if you use that for pain research, and you can use the same microscope that I've been using, then you can look at the activity of neurons without using a dye. Right now, we can use a dye because we can look at a single neuron. But if you want to do live experiments, it will be difficult to use a dye. It will be difficult to use a fluorophore. The best way is to use genetically encoded calcium indicators. And most of the research now is actually going into genetically encoded voltage indicators as well. So you don't just record the calcium, you record also the voltage. Note that I showed you earlier that we need to use an electrode to record the voltage. But a lot of research now is working into using voltage imaging as well, not just calcium imaging.
Okay, thank you very much, Vince. I guess that ends our webinar for today. So, thank you everyone for joining us, and we hope that you will join us in our future webinars. So, have a good day, everyone.